welcome back to ISCT 2024 here in Vancouver, Canada. I am fortunate enough to be interviewing in person our top abstract award winner, Dr. Nisa Renaud,
Yes.
Renaud, from Fujifilm Cellular Dynamics out of Madison, Wisconsin, back in the U.S. So you are the winner of the overall abstract award for this meeting. Congratulations.
Thank you. Very
And as I
excited.
understand, it's your first ISCT meeting ever.
Yes, that's correct.
Way to come out, swinging. That's awesome.
Yeah.
That's great. And so, how are you liking the meeting so far?
It's fantastic.
Yeah.
It's very humbling. It's been just wonderful to learn from people who have a lot of experience with clinical trials and are a little further down that road than I've been able to go yet. So.
Fair enough. This is the place to do it, though. Like, to have those conversations, to meet everyone you want to talk to. Anyone you want to learn anything from is here. So, yeah, that's great. Good for you, taking advantage of it. So I want to talk about your abstract. So your abstract was titled First-in-Man Treatment of Heart Failure by Cardiovascular Cell-Derived Secretome.
Yes.
And so when I first looked at that was kind of a lot to think about. It was quite the complicated manufacturing platform, so I was hoping you could just walk us through kind of how you got to this final product.
Sure. So what the product is that we're exploring in this clinical trial is an EV-enriched secretome, which is coming from cardiovascular progenitor cells, which were differentiated from induced pluripotent stem cells.
Okay.
So our process is from a single donor.
Okay. Like normal skin biopsy or punch biopsy?
Just,
Okay.
normal human cells are reprogrammed to iPSCs, and then we're able to create GMP banks of those iPSCs, and then from a single bank, we're able to differentiate multiple times to cardiovascular progenitor cells, and then we're able to do that all in GMP, and then culture those cells and collect the spent medium and isolate the extracellular vesicles.
How long is that process then?
The process from seeding the iPSCs, doing the differentiation, right through EV manufacture is about 15 days.
Wow, okay.
Yeah.
So, and that's very kind of hands-on, like at every step? Or-
Yes.
Okay.
Yes.
Wow. Oh my gosh. I applaud you for taking them through that. So my next question for that was then, once you've gone through all this and you have your EV-enriched secretome product, when do you collect that, by the way? Like how many days after culture are you taking that?
So they are cardiovascular progenitor cells once we actually start the collection of the secretome.
Yeah.
So we used a two-step process for that aspect. So we have a rich medium that the cells are able to expand a little bit into as CPC. And then we actually flip them to a vesiculation media, which is quite poor in protein and growth factors, so that when we're able to isolate the secretome, it has as little media components kind of coming through that process as well.
Gotcha. Okay, that's smart. Okay, that's great. That would have been my next question because that's really interesting. Okay, so then, all right, you've collected all of that. Then what does release testing look like for a product like that?
So as we've heard a lot from the conference, and I'm really excited that there's a lot of vesicle talk at the conference.
A whole day on it.
It's very exciting, yes. It's a complex mixture of biological molecules, biologically active components. And so the regulatory agencies that we're working with do understand that it is very complex. It's not like a typical antibody production or even like a viral production, which can be very homogeneous. We understand it's very heterogeneous and full of components. So the important thing is to show reproducibility, process control, and then you can show the safety of your product, your final mixture product. So we look at purity, we look at identity, we look at potency assays as well.
A hot topic.
Yes.
Okay.
And then the safety profile. So it's a combination of all of those different aspects, like you would for many products. And as I said, the regulatory agencies do seem to be embracing the fact that it is heterogeneous. It's a mixture of components. So as long as that mixture is the same every time you produce it, then we're doing our job.
Okay. And you said it all came from the same donor, but you can expand multiple times.
Yes, so that's the beauty of iPSCs.
Okay.
That from a single donor, you can have your seed bank, then your master cell bank, working cell banks, so you're not getting that donor-to-donor variability that you would in some other.
What is the variability? I mean, is it like remarkably tight, or even with the same donor, you get some like acceptable variation within the markers?
Yeah, so if you're working from the same cell bank, we do control our process, and we've learned a lot. So speaking from the Fujifilm cellular dynamics experience, we have very controlled processes where your iPSC handling and your differentiation are extremely controlled, and so the product is very reproducible at the end.
That's nice. Okay. Yeah. And then you deliver it fresh to patients, or cryopreserved and then thawed?
That's another beauty of extracellular vesicles, is that they're very thermodynamically stable.
Nice.
So we can freeze them, thaw them very simply. So unlike a cell product where you have to handle them so carefully at the
you'll
on the
lose
day
so
of
much in the
dosing.
freezing and thawing of it. Okay.
Exactly, and it's not alive, so there's no viability testing, at point of view. So it really does simplify that aspect of it.
Sure. Okay. So then you can thaw at bedside and give, essentially?
Yes.
Wow. And it's very off the shelf in that way, too.
Yes, and we were able to produce one giant lot that was able to serve for our GLP animal studies and our entire clinical trial.
So living the dream over
Yes.
there. That's so nice.
Yes.
Okay, so I guess that leads me to my next question. What was the safety profile in the patients when you finally administered it?
Yeah, so I'm really excited to be able to present on behalf of the huge team that has worked on this. That the first patient that has received the 3 doses of secretome, and we've had 1 month and a 6-month follow-up. And the safety profile is looking great. No serious adverse events. The patient is actually doing better in terms of heart failure indices.
Okay.
So very excited, and importantly, this is an allogeneic therapy, so looking for donor-specific antibodies, and there was no evidence of that. So.
Okay, awesome, very promising. That's great. Okay, and it's because the indication you targeted was heart failure, like patients were in active heart failure when they were dosed? Okay.
That's correct. So the patient that I'll be presenting on was in heart failure level 3 when he started in the trial, and now he's down to a level 2, which is fantastic.
And in theory, the patient could keep receiving the therapy like indefinitely.
So it's very different from viral-based therapies or even cell therapy. So there's no immunosuppression that's required, and because there are no donor-specific antibodies, the patient could in theory be redosed. That's not in the plan for right now. So just the 3 doses 3 weeks apart, and the patient continues to improve over time. So. Yeah.
Well, that will be interesting to see, like after the 3-dose course, if they continue the trajectory of. That's awesome. Okay, can't wait to see that. Can you hypothesize what the mechanism of action might be happening?
So that's the $1 million question. Again, because EV and secretome are pretty exciting because they seem to have effects on a lot of different cell types and a lot of different tissues simultaneously. So when we look in the lab at various in vitro assays, we see subtle effects on many different cell types. So it's a mixture of molecules. Even within the vesicle, there is a whole mixture of microRNAs and proteins and lipids, etc. So I think that it is likely to be affecting multiple targets.
So not just the heart, right? So you think you're impacting other things that are then in turn supporting the function of the heart?
Yes. So time will tell as we learn more and more. It certainly seems that there is an impact on the immune system. We've shown that in animal models, and then looking forward to more patient data to come out to see if that comes out in the patients. But definitely inflammation is an important part of heart failure and the progression of heart failure. So if we can reverse some of that negative milieu.
Oh, that's smart. Okay.
Yeah.
I I feel like that's such an emerging area in general, just like
Yes.
how much the immune system is acting in maybe these disease scenarios where you wouldn't have thought so before.
Yes.
But yes, just how much immune modulation can impact positively
Yes.
these different outcomes. That's great. Okay.
So we've definitely seen it in the lab, an effect on immune cells, but also on cardiomyocytes, also on endothelial cells. So the heart is a mixture of many different cell types.
Okay, so that's is that one of the reasons why you targeted this type of therapy in general? Like the fact that you can hit multiple targets with one therapy?
So
Or is it fortuitous?
the reason that we were very interested in the secretome, so when I started this project, I was a postdoctoral fellow in the laboratory of Fernandez-Garcia, who's our collaborator on this clinical trial. So the trial is happening in France. And when I entered the lab, he already had a lot of animal data and patient data on cell therapies. And it was becoming clear in the cardiac field that those implanted cells are rapidly disappearing, and yet the patient's organs continue to improve. So it really evolved out of those early cell therapy studies that we became interested in the paracrine signaling of those therapeutic cell types.
Yeah, but what a good connection to make. Like
Yes.
even after these cells are dying, the patient's getting, or the model is getting better, right? So
Yes.
then why not just hone in on that?
Yes.
Okay, aspect.
And so many advantages to an EV-based therapy.
Yeah. Well, yeah, you've converted me. Now I want, now I want an AV platform in my lab. That's great. All right. Well, we will look forward to hearing more from you and that work. What else are you excited about at the meeting for the rest of the time here?
So we have a big team here from Fujifilm.
Yeah.
Really excited, just the work that we've been able to do with this clinical trial and getting ready for it. We've been developing a lot of platform technologies to try and support other people's vesicle projects.
Oh, cool.
So we're launching a big initiative here at ISCT this year to support people's projects from early discovery right through commercial
That's
manufacturing.
nice. Okay, that's good.
Yeah.
Catch them earlier.
Yeah, exactly.
Prevent mistakes.
Yeah.
Okay, I'll call you early then as I set up my EV platform.
Fantastic.
Great. All right. Well, if people are not able to connect with you at the meeting here in person, how could they get in touch with you after?
Yes. So we have an email address specifically for our EV work that we're doing as a collaborative group. So that would be Fujifilm-EV Solutions with an S at fujifilm.com and reach out, and hopefully we're able to connect and support.
That's great. Yes, hopefully you will get a lot of connections out of this and have a lot more conversations as the conference goes on, because we have such a good crowd here. It's the largest ever at an ISCT conference, so plenty of collaborations to be made. So that's
Awesome.
great. Well, thank you all for watching, and we'll see you next year in New Orleans.